To assemble the TCE Cell One with only Electrolyte, you can start by attaching the electrodes to the current collectors. Check that the springs on the current collectors are still springy, and screw the electrodes on. Just hand tight is fine, and then you can screw the current collector onto each half of the cell body. Again, just hand tight, and then you can put the two cell halves together, aligning the portholes. Use the locking ring to connect the two halves together, and then you can put the plugs in the portholes. You can use a combination of blind plugs and plugs with holes to fill in the electrolyte. Here I'm using 2 blind plugs and 2 plugs that have holes for filling in the electrolyte. You can then take the capillary and attach a syringe, fill the syringe with electrolyte, and then attach the capillary to one of the ports, and push electrolyte into the cell, aligning the cell so that you push gas out and avoid any bubbles inside, and watch for the meniscus of the electrolyte to come out through the other port. And when you see the electrolyte, you can close it with one of the plugs, and you can then unscrew the capillary and close it up. At this point you can take the cell and place it on the cell holder, and that's your assembled cell. To make a measurement with a membrane, after you have attached the current collectors to the cell body, you can place the membrane in the cavity in the cell body. You can then push the two halves together like before to fix the membrane in place and secure the two halves with the locking ring. When you have a membrane in place, you should fill both halves of the cell separately, and that means that you need 4 plugs with holes to fill the electrolyte properly. So in this case, just as before, you can fill electrolyte into the syringe and attach the capillary. And now we will fill electrolyte on this side of the cell, and the electrolyte will come out of this port, so you should orient this upwards to avoid bubbles forming inside. And then you can push the electrolyte through, and just as before, when you see the electrolyte coming out through the exit port, you can close it up, and you can then detach the capillary and fill the other side of the cell in exactly the same way. And when that is done, you can also close up that side of the cell. And when that is done, you have your assembled cell with the membrane in place. To make a 4 electrode measurement, you can attach reference electrodes inside of your cell. To do this, you just have to replace the plugs with the micro reference electrodes that you can insert into the ports. They should be facing opposite each other, and then you can connect the cell in for electrode mode. During this assembly, if something is stuck, you can use the included hex key to loosen these ports. And for the case of the electrodes, you can use the included wrench to loosen these electrodes. These tools should only be used for disassembly, and when assembling the cell, you should only do it by hand and screw these in hand tight.